Hello students, today we see the difference between light microscope and electron microscope. First of all, in light microscope, the energy source is the visible light to illuminate the specimen, whereas in electron microscope, the beam of electrons are used to illuminate the substance. In light microscope, for the magnification purpose, the glass lenses are used. There are objective lenses and eyepiece lenses which are made up of glasses, whereas in electron microscope, the magnetic lenses are used. The source of visible light in case of light microscope is the natural light or the lamp lights, whereas in electron microscope, the tungsten filaments are used. When these heated, they release the electron. Electron microscopes having very much high resolution comparing with the light microscope. Now, what is resolution? It is the capability to see the 2 points separately. In electron microscope, it is a bit expensive, but in light microscope, the cost is very much cheaper. So it is not available, the electron microscope is not available in each and every laboratories or colleges, whereas we can find light microscope everywhere in the colleges, in any institute. In the light microscope, we can see the normal image, whereas in electron microscope, the detailed image have been found. Even the 3D images are found. In case of light microscope, the color we see may be different due to the use of the variety of stains, whereas in electron microscope, only the black and white images are found. It is very much difficult to carry electron microscope, but in case of light microscope, it is very much handy, so we can use it easily. In electron microscope, the cellular organelles are easily found, and we can distinguish among the organelles. But in light microscope, only the general structure of the organelles, only the general structure of the cell is visible, not we can differentiate among the organelles. Like in nucleus, we can see only the dots. So these are the differences between them.